Good morning, everyone. I am delighted to welcome you all today. Today, we will be exploring a very important topic, the protocol for validation of analytical methods. My name is Dr. S.K. Singh, and I hold a PhD in this field. In this presentation, I will guide you through the key steps and best practices involved in validating analytical methods. We'll look at why validation is crucial, the procedures involved, and how to ensure your methods are robust and reliable. So, let's get started and delve into this essential aspect of analytical science. Let's start by understanding what method validation really means. It's the process through which we demonstrate, using documentary evidence, that our analytical method performs as expected for its specific purpose. Essentially, we want to make sure that the method's performance characteristics, like accuracy, precision, and specificity, meet the requirements for the particular application we have in mind. It's important to remember that any analytical method used to analyze samples must be validated. This isn't a one-time task. It's a critical step to ensure reliability. Plus, the suitability of these test methods should always be verified under the actual conditions where they'll be used, and this verification needs to be well documented for future reference. Moving on to the validation process itself, a well-planned approach is key. We need to follow a structured set of steps to ensure thorough validation. One of the first and most important steps is to develop a validation protocol. This protocol should be a clear, written document that everyone involved can understand and approve. It should include detailed instructions, laid out step by step, so that the process is consistent and repeatable. For example, when validating an HPLC method, you'd create a protocol that specifies all the parameters, conditions, and acceptance criteria. The good news is, the same principles generally apply across different instrumental test methods. So once you master preparing these protocols for one technique, it becomes easier to adapt them for others. In summary, developing a solid validation protocol is foundational. It guides the entire process and ensures that our method validation is thorough, consistent, and aligned with regulatory expectations. Let's start with the typical method validation protocol, which is a crucial part of our overall quality assurance process. First, we have the introduction, where we outline the firm's validation policy and provide a general description of our approach. This sets the stage for understanding how we ensure consistency and reliability in our methods. Next, we look at the organizational structure. Here, we describe who is responsible for what, detailing all personnel involved in validation activities. Clear responsibilities help us maintain accountability and streamline the process. Then, there's the process and product description. We give a brief overview of the process itself and the product involved, or refer to the relevant documents that provide this information. This helps ensure everyone is aligned on what exactly is being validated. We also include specific process considerations, which highlight the critical characteristics of the process. These are the key factors that could impact the validity and quality of the process and must be carefully monitored. Following that, we define the key acceptance criteria. This is where we establish the general standards or benchmarks that the process must meet to be considered successful. The documentation format is also covered, describing the structure and format used for the protocol and the final report. Consistent documentation is essential for regulatory compliance and future reference. We then list the required SOPs, standard operating procedures, that are relevant to the validation process. These SOPs guide the activities and ensure consistency across validation efforts. Planning and scheduling are next, where we detail the resources, equipment, and chemicals needed, along with a timeline for the project. Proper planning helps us stay on track and ensures all elements are ready when needed. Finally, change control is addressed. This involves describing or referencing how we handle variations in critical process parameters, whether they relate to the process itself or the product. Having a solid change control system is vital for maintaining validation integrity over time. All of these elements together form a comprehensive approach to method validation, ensuring our processes are robust, reproducible, and compliant with regulatory standards. Now, let's turn our attention to the analytical method validation protocol, starting with the cover page. This page serves as the official introduction to the document, clearly indicating the purpose and scope of the validation process. It typically includes essential details, such as the method being validated, the laboratory or organization conducting the validation, and relevant dates. The cover page is important because it ensures that everyone involved is on the same page right from the start. It also provides a professional and organized appearance, setting the tone for the thoroughness and rigor of the validation process that follows. When reviewing validation protocols, always check this cover page to confirm the method and scope are aligned with your project or regulatory requirements. Now let's take a closer look at the validation parameters. The analytical methods we need to validate are quite diverse, each serving a specific purpose. First, we have identification tests, which are essential to confirm the true identity of an analyte. This step ensures we're analyzing the correct substance and that it meets the required specifications. Next, there's the quantitative test for impurities. This method must accurately reflect the purity of the sample, meaning it should precisely measure how much impurity is present. This is crucial for quality control and ensuring safety. Then we have the limit test for impurities. Unlike the quantitative test, this one is more about establishing whether impurities are below a certain threshold. It provides a quick indication of the purity characteristics of the sample. We also need to validate assay methods for drug substances and drug products. These are designed to measure the amount of the active ingredient present. It's important that these methods are both accurate and quantitative. Plus, they often include analysis for content uniformity and measuring analytes from dissolution samples, which are critical for ensuring consistent product quality. Finally, all these methods have specific characteristics that need validation, and we'll see a summary of these in the following table. Validating these parameters ensures our analytical methods are reliable, consistent, and fit for purpose, which ties directly into our overall goal of delivering safe and effective pharmaceuticals. Now, let's discuss the key aspects of analytical performance characteristics, starting with specificity. Specificity, or selectivity, refers to our method's ability to accurately identify our analyte even when other compounds are present. These could be impurities, degradants, or components from the matrix that might interfere. Ensuring specificity involves separating the analyte from these potential contaminants. We typically assess this through chromatographic separation, looking at the peaks for purity. A peak purity test helps us confirm that no other substances are co-eluting with our analyte. To do this, we often use detectors like PDA or MS. When reviewing chromatograms, we include labeled peaks and report parameters like resolution, plate count, and tailing factor in our validation report. Next, the test procedure involves injecting a prepared extract of the placebo, which helps us verify that excipients or other non-target components do not interfere with detecting our analyte. We also document this process by printing chromatograms for record keeping. Finally, the acceptance criteria are straightforward. Excipient compounds should not interfere with our target analyte's detection. If these points are met, we can be confident in our method's specificity, which is crucial for the reliability of our analytical results and the overall quality of our product. Let's talk about linearity, which is a crucial aspect of method validation. Linearity refers to a method's ability to produce test results that are directly proportional to the concentration of the sample within a specific range. For HPLC methods, this means that the detector response, such as peak area or height, should have a straight-line relationship with the sample concentration. We can demonstrate this linearity directly on the drug substance, either by diluting a standard stock solution or by weighing the components separately, following the established procedures. To evaluate linearity, we typically start with a visual inspection, plotting the signals against known concentrations. If the plot looks linear, we then confirm this with statistical methods, like regression analysis, which provides a mathematical measure of how well the data fit a straight line. The regression line gives us important information, specifically the degree of linearity, often expressed as the variance around the slope. In some cases, the response might need to be described by a specific mathematical function of the analyte concentration. And of course, different pharmaceutical methods have established linearity ranges and acceptance criteria, which are summarized in the following table. Overall, ensuring linearity helps us confirm that our method can reliably quantify samples across the desired concentration range, which is fundamental for accurate and consistent results. Let's walk through the test procedure. We start by preparing standard solutions at 6 different concentrations, typically at 25, 50, 75, 100, 150, and 200% of the target concentration. For each of these levels, we analyze 3 independently prepared replicates. It's important to note that the method used for preparing these standards and the number of injections will be the same as in the final method, ensuring consistency. Next, we need to document all results carefully on a datasheet. For each concentration, we calculate the mean response, the standard deviation, and the relative standard deviation, or RSD. Then, we plot the concentration on the x-axis against the mean response on the y-axis for all levels. This visual helps us assess linearity across the range. We also derive the regression equation and determine the coefficient of determination, which is the R-squared value. Recording these calculations on the datasheet is essential for later analysis. Moving on to acceptance criteria, the correlation coefficient, or R, for these 6 concentration levels should be at least 0.999, especially within the range of 80 to 120% of the target concentration. The y-intercept of our regression line must be less than or equal to 2% of the target concentration response. Additionally, if we plot the response factor versus concentration, all the values should fall within 2.5% of the target response factor within the 80 to 120% range. This ensures the method's accuracy and linearity. For the overall performance, the coefficient of determination should be at least 0.997 for active ingredients, 0.98 for impurities, and 0.95 for biologics. These criteria help us confirm that our method is reliable, precise, and suitable for quality control and validation purposes across different types of compounds. Now, let's talk about the range of an analytical method. The range is essentially the interval between the lowest and highest concentrations of the analyte in a sample for which we have proven the method works reliably. This includes both ends of the spectrum. It's important because it shows us where the method maintains acceptable levels of precision, accuracy, and linearity. Typically, we determine this range through linearity studies, and it depends on what we intend to do with the method. For example, in assay methods, the usual range covers from 80% to 120% of the test concentration. So, if you're testing a drug, your method should be reliable within that span. For content uniformity, the range is broader, generally from 70% to 130%, which makes sense because uniformity testing might involve more variability depending on the dosage form. When it comes to dissolution testing, the acceptable range is about plus or minus 20% over the specified range. And for impurity determination, the range starts from the impurity reporting level up to 120% of the specification limit. The key point here is that a method's range is only confirmed when it meets the necessary criteria for linearity, accuracy, and precision throughout that span. So, understanding and properly establishing this range ensures the method is suitable for its intended application. Now, let's talk about the test procedure. The data we gather during the linearity and accuracy studies will help us determine the operational range of our method. Basically, we'll look at how well our method performs across different concentrations to ensure it's reliable. The precision data, which comes from analyzing 3 replicate samples at each level during the accuracy studies, is also crucial here. This helps us understand the consistency of our measurements within that range. When it comes to documentation, it's important to record the range directly on the datasheet so everything is clear and traceable. As for the acceptance criteria, the acceptable range is defined by a couple of key points. First, it must show linearity and accuracy within the specified criteria we've set earlier. Second, and very importantly, the precision within this range should be quite tight, specifically a relative standard deviation, or RSD, of 3% or less. So, to sum up, we're looking for a concentration interval where our method is both accurate and linear, and where the measurements are consistently precise. This ensures the method is robust and reliable for routine analysis. Let's talk about accuracy in analytical methods. Accuracy essentially measures how close our obtained value is to the true or reference value, which we accept as the standard. Now, in practice, no measurement process is perfect, so we can never know the true value exactly. This means we often assess accuracy by analyzing samples with known concentrations. For example, we might spike a sample with a known amount of analyte and see how much we recover. Alternatively, we compare our results to certified reference materials that have a known purity. If a placebo isn't available, we can use the standard addition method, where we add a known amount of analyte directly to the sample. When checking for impurities, we analyze samples with a known amount of impurities. To get reliable accuracy data, we should perform at least 9 measurements across 3 different concentration levels, with 3 replicates each. This helps us cover the entire range we're interested in. We typically report accuracy as the percentage recovery, how much of the known added analyte we detect, or as the difference between our mean result and the true value, along with confidence intervals for statistical confidence. It's important that the concentration range we test reflects the real-world range of concern. Recovery can vary depending on the sample matrix, processing procedure, and analyte concentration. For drug substances and products, the acceptable accuracy range is generally between 98 and 102%, and 97 to 103%, respectively. For impurity analysis, a broader range of 50 to 150% of the average recovery is acceptable. Overall, understanding and verifying accuracy is crucial because it underpins the reliability of our analytical results, which is the core theme of our discussion today. Now, let's talk about the test procedure. We prepare spiked samples at 3 different concentrations, ranging from 50 to 150% of the target concentration. For each of these concentrations, we analyze 3 separate replicates. If it's challenging to prepare known placebos, we can instead use a low concentration of a known standard to serve as a baseline. Moving on to documentation, for each sample, it's important to report the theoretical value, the actual assay value, and calculate the percent recovery. We then determine the mean, standard deviation, relative standard deviation, and overall percent recovery across all samples. All these results should be carefully recorded on the datasheet for traceability. Now, regarding acceptance criteria, the goal is that the mean recovery falls within 90 to 110% of the theoretical value for non-regulated products. In the pharmaceutical industry, a typical target is around 100 plus, or minus 2%, for an assay of an active ingredient within a range of 80 to 120% of the target concentration. Keep in mind, lower recoveries can sometimes be acceptable depending on the method's needs. But the key point is that the accuracy requirements include a bias of no more than 2% for dosage forms and 1% for drug substances. This ensures our method is both precise and accurate, which is essential for reliable analysis and quality control. Let's talk about precision in analytical methods. Precision essentially tells us how close our measurements are to each other when we repeat the same test under the same conditions. It's a key aspect of method validation because it shows us the consistency of our results. We can think of precision on 3 levels: repeatability, intermediate precision, and reproducibility. Repeatability, also called intra-assay precision, is about how consistent our results are when we test the same sample multiple times in a short period, using the same equipment and operators. For example, performing 6 determinations on a 100% concentration sample or testing 3 samples at different concentrations in triplicate. Moving on to intermediate precision, this looks at variation within the same laboratory but over different days, with different analysts, or even using different equipment. It's also known as ruggedness, and it helps us understand how reliable our method is under typical lab variations. To evaluate this, we usually prepare multiple samples and standard solutions across different days or conditions. Finally, reproducibility assesses how well our method performs across different laboratories. This is often tested during collaborative studies, especially when transferring a method from one lab to another. It involves interlaboratory trials to ensure the method's robustness and transferability. When we analyze our precision data, we typically express it through measures like standard deviation, RSD, and confidence intervals. For major analytes, an RSD of 2% or less is generally considered acceptable. However, for low-level impurities, an RSD between 5 to 10% is acceptable, reflecting the inherent variability at those lower concentrations. Understanding these different levels of precision helps us ensure our analytical methods are reliable, consistent, and fit for the purpose, which is fundamental in maintaining the quality and integrity of our results in the overall analytical process. Now, let's talk about the precision and repeatability test procedure. First, we prepare one sample solution that contains the target level of analyte. From this single preparation, we make 10 replicates, following the final method procedure carefully. This helps us assess how consistent our method is when repeated under the same conditions. Next, during documentation, it's important to record key data points like the retention time, peak area, and peak height for each replicate on the datasheet. Once we have these, we calculate the mean, standard deviation, and relative standard deviation, or RSD, to understand the variability in our measurements. Moving on to acceptance criteria, the RSD should be 1% for drug substances, which indicates very good precision. For drug products, a slightly higher RSD of 2% is acceptable. When it comes to minor components, an RSD of about plus or minus 5% is typical, but it can go up to 10% at the limit of quantitation. Remember, maintaining these precision standards is crucial because it ensures our method produces reliable, reproducible results, which ties directly into the overall quality and consistency of our analytical process. Now, let's talk about intermediate precision, which is also known as within-laboratory variation. To demonstrate this, we have 2 analysts working with 2 different HPLC systems on separate days. They will evaluate the relative percent purity data at 3 concentration levels: 50%, 100%, and 150%. These levels are chosen because they cover the typical assay range of 80 to 120%, ensuring our method is reliable across the spectrum. As part of the process, it's important to document everything carefully. For each concentration, record the relative percent purity, or percent area, on the datasheet. Then, you'll want to calculate the mean, the standard deviation, and the relative standard deviation, or RSD, for both operators and instruments. This helps us understand the variability within our lab. The key acceptance criterion here is that the assay results from the 2 operators, using the 2 different instruments on different days, should have an RSD of 2% or less. This indicates that our method is precise enough for routine use. In summary, this step confirms that our analytical method produces consistent results across different analysts and equipment, which is crucial for ensuring reliability and reproducibility in our testing process. Let's talk about the limit of detection, or LOD, which is a crucial concept in analytical procedures. Essentially, it refers to the smallest amount of analyte in a sample that we can detect, though not necessarily quantify precisely. Now, there are a few ways to determine this detection limit. The simplest method relies on the signal-to-noise ratio. In this approach, we compare the signals from samples with a known low concentration of analyte to those from blank samples, which contain no analyte. When the signal is about 3 times the noise level, or sometimes 2 times, we consider that as an acceptable detection limit. Another way involves looking at the standard deviation of responses and the slope of the calibration curve. Here, the detection limit can be expressed mathematically as LOD equals 3.3 times sigma divided by S, where sigma is the standard deviation of the response and S is the slope of the calibration curve. To estimate the slope, we typically use the calibration curve generated from samples with known analyte concentrations. The standard deviation, sigma, can be estimated from the blank samples or directly from the calibration curve, especially if we analyze samples within the detection range. We often use the residual standard deviation of the regression line or the standard deviation of the y-intercept to get an accurate estimate. Finally, there's also a visual evaluation method, which is particularly useful for non-instrumental techniques. Although less common for instrumental methods, it can still provide valuable insights. Overall, understanding and accurately determining the LOD helps us ensure the reliability of our analytical methods and the integrity of our results. Now, let's talk about how we determine the limit of detection, or LOD. Essentially, the LOD is established by analyzing samples with known concentrations of the analyte. We look for the minimum level at which we can reliably detect the analyte, which means the signal stands out clearly from the background noise. The chromatograms we generate during this process are key. They provide visual proof and justification for the detection limit we set. Moving on to the test procedure, we start by finding the lowest concentration of our standard solution. We do this through sequential dilutions, gradually decreasing the concentration until we reach the point where detection is just possible. From this diluted sample, we prepare 6 replicate measurements to ensure consistency and reliability. Once we've run these tests, we document everything carefully. We print out the chromatograms and record the lowest concentration at which the analyte is detectable. We also note the RSD, or relative standard deviation, to assess the precision of our measurements. Finally, the acceptance criteria for the detection limit is a signal-to-noise ratio of 3:1. This means the signal of the analyte must be at least 3 times greater than the background noise to be considered reliably detected. This ratio helps us maintain confidence in our detection capability and ensures the method's robustness. Now, let's move on to the limit of quantitation, or LOQ. This is essentially the smallest amount of analyte in a sample that we can measure with acceptable precision and accuracy. Think of it as the lower threshold where our analytical method still produces reliable results. The LOQ is primarily influenced by 2 factors: the sensitivity of our detector and how accurately we can prepare our samples. These factors determine how well we can detect and quantify low concentrations of analytes. Just like with the detection limit, we can determine the LOQ using a similar approach. However, for LOQ, we look for a signal-to-noise ratio of 10:1, meaning the signal must be 10 times the background noise. The calculation for LOQ involves the standard deviation of our response, denoted as sigma, and the slope of our calibration curve. The formula is LOQ equals 10 times sigma divided by S. In practice, we estimate these values just as we do for the detection limit. Sometimes, the LOQ can also be established visually, based on what we observe in our data as the lowest reliably quantifiable concentration. It's important to remember that at the LOQ level, our analyte concentration should be measurable with acceptable accuracy and precision. Typically, we accept a mean recovery between 50 and 150%, with a relative standard deviation of 25% or less. This ensures our method is robust enough to quantify low levels of analyte accurately, which is essential for reliable analytical results across various applications. Now, let's talk about the test procedure for determining the limit of quantitation. The goal here is to establish the lowest concentration of an analyte in the sample matrix that we can measure accurately and precisely, according to the method's requirements. Often, this value corresponds to the lowest point on the standard curve. Once we've identified that concentration, we make 6 replicates from this solution to verify consistency and reproducibility. Next, we need to document everything carefully. This means printing out the chromatogram, recording the lowest quantified concentration, and noting the relative standard deviation, or RSD. This data helps us demonstrate that our method meets the necessary accuracy and precision criteria for acceptance. Now, moving on to the acceptance criteria. The limit of quantitation, or LOQ, is typically defined in chromatographic methods as the concentration that produces a signal-to-noise ratio of at least 10:1. In other words, the peak should be at least 10 times higher than the baseline noise. The LOQ is essentially our best estimate of a low concentration, where we can still reliably measure the analyte, with an RSD close to 10%, based on a minimum of 6 replicate determinations. Understanding these concepts is crucial because they ensure our analytical method is sensitive enough to detect and quantify low levels of analytes accurately, which is fundamental to the overall integrity of our testing process. Let's talk about robustness, which is a key aspect of an analytical procedure. Essentially, robustness measures how well the method can withstand small, deliberate changes in its parameters without losing accuracy or reliability. During method development, evaluating robustness gives us an idea of how dependable the method will be during routine use. The goal here is to identify the critical operating parameters that are essential for the method's success. Once identified, these parameters should be carefully controlled, and any necessary precautions should be clearly documented. In the context of HPLC, for example, we look at parameters like pH, flow rate, column temperature, and mobile phase composition. We vary these within a reasonable range to see how they affect the system's performance. The key is to analyze the system suitability parameters under each condition to identify which factors significantly influence the method's accuracy and precision. We also consider the stability of the analytical solution and the extraction time as part of the robustness study. For stability, we test how long the analytical solution remains reliable by checking its results after 4, 12, 24, or even 48 hours. The criteria for stability are based on the relative difference between the initial measurement and the measurement after the specified time. For drug substances and drug products, a difference of 2% or less is acceptable, while for impurity testing, up to 10% difference can be tolerated. In summary, robustness helps ensure that our method is reliable and consistent under normal working conditions. It's about understanding which parameters are critical and making sure they are properly controlled, so we can trust our results in routine analysis. When we talk about filtration in sample preparation, it's important to understand that filter paper studies can be quite useful. By filtering the sample solution through different types of filter paper, we can analyze how each one performs, which helps us choose the best option for our method. Moving on to chromatography, when we analyze a sample that contains some impurities, we often modify certain parameters to see how the results change. The idea is to compare the chromatography results obtained under these modified conditions with those from the original target parameters. Specifically, we look at changes like adjusting the methanol content in the mobile phase by plus or minus 2%, shifting the mobile phase pH by plus or minus 0.1 pH units, and increasing or decreasing the column temperature by about 5 degrees Celsius. The goal here is to see if these small variations still produce acceptable, reliable chromatography results. If they do, then these conditions can be incorporated into the final method. This process is a key part of robustness testing, which helps us ensure that our method remains consistent even when small, normal variations occur during routine analysis. It's all about making our method more reliable and flexible without sacrificing accuracy. Let's talk about system suitability, or SSD, which is a crucial part of many analytical procedures. Essentially, SSD helps us verify that our equipment, the analytical operations we perform, and the samples we're working with are all functioning properly as a system. It's based on the idea that these components are interconnected and can be evaluated together. The main purpose of conducting a system suitability test is to give us confidence that, on a specific day, our method is producing accurate and precise results. This isn't a one-time check. We run these tests every time we use a method, either before starting the analysis or during the process itself. When we perform an SSD, we compare the results to predefined acceptance criteria. If the results meet these criteria, we can be assured that the method is satisfactory for that particular occasion. For example, in HPLC methods, SSDs confirm that the system is ready and capable of performing the analysis reliably on a daily basis. Some of the primary parameters we look at include resolution, which tells us how well two peaks are separated, repeatability, measured by the % RSD of peak response and retention time, column efficiency, indicated by the N value, and the tailing factor, TF, which shows peak symmetry. We also consider parameters like the retention factor, K, and the separation factor, alpha. Finally, there are specific limits for these parameters, which are listed in a table that guides us on whether our system is performing adequately. Overall, SSD provides that added layer of assurance, ensuring our analytical method remains robust and reliable over time. Now, let's talk about the test procedure for system suitability. We will perform these tests on both HPLC systems to ensure they are working accurately and precisely. To do this, we'll inject 6 times a solution that contains the analyte at 100% of the test concentration. By doing this, we can evaluate several important parameters. These include the plate count, which indicates the efficiency of the column, the tailing factors, which tell us about peak symmetry, the resolution, to see how well the peaks are separated, and reproducibility, which we measure by calculating the % RSD of retention time, peak area, and peak height across those 6 injections. Next, once we've done these injections, it's crucial to document everything properly. So, we print out the chromatogram and record all the data on the datasheet. Proper documentation ensures traceability and helps us verify that the system is performing adequately. Now, let's discuss the acceptance criteria. First, the retention factor, or K, should be at least 2. This means the peak of interest needs to be well resolved from other peaks and the void volume. Resolution, RS, should be 2 or greater between our target peak and any nearby interfering peaks, like impurities or degradation products. For reproducibility, the RSD for peak area, height, and retention time should all be within 1% across the 6 injections. The tailing factor, T, should be around 2, indicating acceptable peak symmetry. And finally, the number of theoretical plates, N, should be at least 2,000, which signifies good column efficiency. In summary, these parameters and criteria are vital to confirm that our HPLC system is performing reliably and consistently, ensuring the quality of our analytical results. Revalidation is a critical part of maintaining the integrity of our analytical methods. It becomes necessary whenever we make changes to a method, especially if those changes push parameters outside the initially defined operating range. It's important to specify the operating parameters clearly, including their ranges, so we know exactly when revalidation is needed. For example, if we discover a new impurity during analysis that affects the method's specificity, the method may no longer be suitable and will require modification and revalidation. Similarly, any changes in equipment or the chemical quality of reagents can impact the method's performance. Therefore, such changes also trigger the need for revalidation to ensure continued accuracy and reliability. Now, when it comes to pharmacopoeial methods, the process is a bit different. These are validated methods listed in official pharmacopoeias, so users don't need to validate them from scratch. Instead, they perform a verification to confirm these methods work well under their specific conditions and for their particular drug substances or products. Verification isn't about repeating validation. Rather, it's about assessing key performance characteristics, like accuracy, precision, or specificity, to gather objective evidence that the method is suitable for its intended use. This step helps us ensure that the methods are fit for purpose without unnecessary repetition of validation, aligning with the overall goal of ensuring consistent quality and reliable results. If the verification of the compendial method is not successful, it may be concluded that the method might not be suitable for use with the specific article being tested in that laboratory. In such cases, an alternative method, which is allowed in the general notices, can be developed and validated instead. Verification requirements should be tailored based on an assessment of the method's complexity and the material to which it's applied. Only those characteristics that are relevant and appropriate for verifying the particular method need to be evaluated. It's important to note that full revalidation of the original compendial method is not usually necessary just to verify its suitability under actual testing conditions. However, some key analytical performance characteristics like accuracy, precision, and specificity can be used during the verification process. Verification should also include an evaluation of factors such as how the matrix affects the recovery of impurities and active substances from the drug product. Additionally, it should assess the adequacy of chromatographic conditions, the suitability of the column, and the response of the detector signal, among other things. Overall, this process helps ensure that the method is fit for purpose and reliable for testing, which is crucial for maintaining quality and compliance in our laboratory work. For further support, please don't hesitate to contact us. We're here to help clarify any questions or provide additional resources you might need. Remember, our goal is to ensure you feel confident moving forward with this information. Thank you so much for your attention and participation throughout this presentation. We truly appreciate your engagement and look forward to supporting you in your next steps.